Hello all, today I am here to discuss with the topic post-transcriptional modification or processing of eukaryotic pre-mRNA. So let's start. mRNA processing is the structural modification and functional maturation of newly synthesized mRNA molecules. It is an essential prerequisite for mRNA functioning and gene regulation. The modification occurring during transcription is called co-transcriptional modification, and the one taking place after the completion of transcription is called post-transcriptional modification. An mRNA molecule copied or transcribed from a DNA template is called mRNA transcript, and it is the primary mRNA transcript. It is an exact replica or negative copy of the DNA template strand from which it has been transcribed. The processing of eukaryotic pre-mRNA involves five major events, namely modification of bases, splicing, capping, tailing or polyadenylation, mRNA editing. Modification of bases. The chemical modification of bases is accomplished by the methylation of interior adenine residues. Splicing. It is essentially the removal of the non-coding regions and the subsequent ligation of the coding regions of the mRNA. During this, the non-coding regions and the introns that are not required in the genetic translation are cleaved out or sliced away by the ribonucleases. The remaining exons are soon ligated or joined together by the enzyme RNA ligase. Thus, the excessively long pre-mRNA is trimmed to a stable molecule of desired length and required nucleotide composition. The splicing out of the introns involves the excision or cleaving of the phosphodiester bonds, whereas the ligation of exons occurs through phosphodiester bonding. Excision and ligation must be highly precise because the addition or loss of even a single nucleotide will abnormally alter the reading frame of the message. Capping. Capping is the formation of the protective cap at the 5' end of the pre-mRNA by the addition of 7-methylguanosine. It may begin even before transcription is over, so it is a co-transcriptional as well as the post-transcriptional process. It involves two major events, namely methylation of the first two or three nucleotides at the 5' end and the capping of the molecule by the incorporation of an inverted 7-methylguanine nucleotide phosphate residue with the terminal nucleotide through the unusual 5 to 5' triphosphate linkage using GTP. Capping is catalyzed by the enzyme mRNA guanine transferase. Tailing or polyadenylation. It is the process of polyadenylation or incorporation of numerous adenine nucleotide at the 3' end soon after transcription. As many as 200 or more adenine nucleotides are added with the help of the enzyme adenylic acid transferase. Capping and tailing are believed to protect the mRNA from the degradative actions of ribonucleases. Apart from this, its polyadenylate tail may possibly help to pass through the nucleopore to the cytoplasm. mRNA editing. It is a mechanism which alters the genetic information encoded by the exon sequences in mRNA. It involves the precise addition and deletion of nucleotides within the coding regions of mRNA. Thus, in turn, it modulates the genetic activity. mRNA editing results in an mRNA molecule which is different from its DNA template with respect to the nucleotide sequence in the coding region. This difference is not due to the splicing out of the introns but due to the addition, deletion, or rearrangement of nucleotides. RNA editing can be co-transcriptional or post-transcriptional. The former occurs simultaneously with the transcription, but the later only after the completion of transcription. The genes subjected to the RNA editing are called cryptogenes. In mammals, RNA editing is believed to determine the properties of some ion channels and G protein coupled receptors. The evolutionary significance of RNA editing is not definitely known, but there are four kinds of mRNA editing. First one, symbol editing. Second one, insertional editing. Third one, pan editing. And fourth one, polyadenylation editing. Symbol editing is a conversion of only a single nucleotide. It is a post-transcriptional process. Insertional editing, it is a co-transcriptional insertion of either a single nucleotide or short runs of nucleotides. Pan editing, it is a post-transcriptional insertion or deletion of multiple nucleotides or the addition of deletion of multiple uracils or cytosine nucleotides. Polyadenylation editing, post-transcriptional addition of adenine nucleotides at the 3' end of the mRNA transcript. So, happy learning and thank you.